this death in all around the world, and millions of people are daily dying. And as a matter of fact, the very efficient compound artemisinin was discovered in 2015 by the Chinese scientists, and it is actually trying to overcome the death problem with the malaria. As a matter of fact, artemisinin is one of the major compounds produced by the plant Artemisi annua, and as you can see, the pathway, the biosynthetic pathway that produces this compound has been well studied, and several enzyme-coding genes have been elucidated and they have been found, and a lot of efforts have been focused on making the overexpression as well as the overproduction of artemisinin in this plant. However, there are basic challenges when we are dealing with this artemisinin. The first biggest challenge is the rising resistance in the parasites of malaria, which is the Plasmodium species. So what the scientists are doing, they are suggesting to start artemisinin combinational therapy, which means combining artemisinin with other compounds probably can reduce the resistance rate in the Plasmodium species. And beside that, the second challenge about artemisinin is that the production rate in this plant is quite, in a natural phenomenon, it's really low. So what they are doing is genetic engineering of the plant, including the enzyme-coding genes, overexpression, as well as transcription regulation are the two key, most important techniques to overcome the lack of the artemisinin compounds in this. So in case of the combination therapy, there are several researchers suggesting that there are other compounds in the plants, including flavonoids, that they can play a key role in combination with artemisinin to overcome the resistance in that. So in that case, I have elucidated two basic pathways of artemisinin biosynthetic as well as the flavonoid biosynthetic pathways. So to see if there is a possible linkage, to find there is any linkage possible between these two distinct pathways. The flavonoids and artemisinin, there are completely two different pathways in the plants. But as a matter of fact, the regulation of them, if you go through the promoter regions of the enzyme coding genes of these two pathways in this plant, you will see that there are mutual binding boxes in the promoter regions of these two pathway genes. And the major families that are well studied and well known to control the regulation of these enzymes have been well studied. As you can see, they have been well distributed all around the promoter regions of these genes. And among all these families, the NEAT transcription factor has shown the promising presence in the promoter region of almost all these genes. So, for that case, I have nominated two MIBs, and I call them MIB1 and MIB2, and I started to study the possible linkage between the flavonoid and artemisinin pathways by overexpression techniques to see if I can increase artemisinin and flavonoid contents in the transgenic plants. Besides that, there is also a possible linkage of these two pathways at the structural genes level, which means the enzyme coding genes for the two pathways, they may play a role, by overexpression, they may play a role in enhancing the production of artemisinin. For that case, we can see the gene expression profile that I have presented here. The top four, they are the artemisinin pathway genes, and the last genes are the flavonoid pathway genes. As you can see, the expression pattern in the trichome, where is the main section of the plant that artemisinin is produced in there, is showing promising results for the PAL, CHI, as well as F3'H. So these might suggest that these genes can be also a good candidate for overexpression strategy. But for this case, the first step of my experiment to find out the possible linkage between the MIB1 and the genes from the artemisinin pathway, including CYP, ADS, ALDH1, as well as DBR2. The dual luciferase results clearly indicated that the MIB1 is capable of enhancing the expression of ADS as well as ALDH1. And in case of MIB2, there was no significant difference showing, so that's why the MIB2 was not counted for the overexpression strategy. But for the MIB1, it was good candidates. Therefore, at the same time, I started to find out if there is any linkage between the MIB1 and MIB2 with the three genes of the flavonoid biosynthetic pathway, including CHI, CHS, and DFR. So as you can see in the figures, as you can see in the sample average, including CHI, CHS, and they are showing the high activity, dual luciferase activity, when they are combined with the MIB1. And in case of the MIB2, they are not showing much differences, so very slightly activation of the promoter regions. So probably this MIB1 could be a good candidate gene for overexpressing strategies. For that case, we constructed the overexpressing vector and we got the transgenic plants. At the first step, what we did, we checked the expression level of these key enzymes, including CHS, CHI, and DFR, which are the enzymes of the flavonoid engine, as well as ADS, CYP, DBRT, and ALDH1, which are the artemisinin pathway. So the results from the qPCR were consistent with the results from the luciferase. So as you can see, the CHS, CHI, and DFR, they perfectly overexpressed. I mean, they had a higher expression level, but in case of the artemisinin pathway genes, ADS, DBRT, and especially ALDH1, they were showing high expression level in overexpressed MIB1 plants. The next step was to find if there is any possible linkage to find out the binding boxes of this MIB on the promoter region of this gene. So for that case, the constructed Welter P-Laxi was constructed with the binding box in the promoter regions of CHI as well as DFR. And as you can see, my PMSA results clearly indicated that this MIB protein is capable of binding to the MB1 boxes of the flavonoid pathway genes. However, the results, they did not clearly indicate it. They did not show any direct binding of this MIB with the promoter regions of artemisinin pathway. So probably the next step for doing this experiment to find out the binding sites of this MIB with other, trying to find out if there is any indirect interaction between this MIB and the promoter region of artemisinin pathway gene. For that case, the yeast-2 upgrade assay was conducted. Okay, and we found out that the MIB2, this MIB is not showing any interaction with a well-known transcription factor regulating artemisinin pathway. So this step was failed and we could not get any interaction. However, there are other members of the transcription factor that they might have interaction with. And you can see clearly that HPLC analysis in transgenic plants show the highest amount of artemisinin in my E line, and as you can see, 26.18, which is 2.6-fold higher than the control. Not only about artemisinin content, but also DHAA and artemisinic acids, it was showing higher content in transgenic plants. The second part of the project was to simultaneous co-transformation of 5 enzymes, including 3 enzyme coding genes, including HMGR, DBR2, and FPS, and 2 transcription factors which are trichome specific, including HD1 as well as ORA. So we constructed 3 independent vectors. The first vector, as you can see, is harvesting 3 genes of the artemisinin pathway, and 2 other vectors are harvesting ORA, which is a transcription, which is a glandular trichome specific, and HD1. As you can see, we have these results here. The results from the T0 and T1 generations, it showed promising results in the line E and line F. In the T0, it increased the artemisinin content up to 2.6-fold. And in case of T1, you can see it is also showing a higher content. So probably this transformation strategy was pretty successful. And at the same time, we checked the overexpression results of this gene because we don't have time. I just quickly pass through this. And we show that all these genes that we try to overexpress, it's showing a higher expression level in transgenic plants in T0 as well as T1 lines. So, and an important thing which was about the trichome density, because ORA and HD1 are trichome specific, and we believe that overexpression of these genes can enhance our trichome density. So as you can see in a plant, experimental plant, transgenic plant, it is showing the higher amount of trichome. As you can see, the pictures also clearly stated that they are showing the higher trichome density. So the most important thing about this that the results have been already reported that transformation of these genes, these 5 genes individually can increase artemisinin content. But in combination together, it enrich the amount of artemisinin content up to 3.5-fold higher than the control, which possibly suggests that this might be a better candidate cascade for a combination for co-transformation strategies. And some of the achievement that I did in this postdoc period was instructing the foreign students for 2017 Foreign Students Affair, and also the award by the Certificate of Professional and the Technical Positions in Shanghai Jiaotong University, attending the oral presentation in 2016 ABBS conference, and mentoring some of the PhD students here. And these are the recent publications that I have submitted. The first paper, which has been submitted to the Trends in Plant Science, and it's just under the final revision, and probably we get the good results soon. And the second paper, it's acted by Chemical and Biophysical Clinical. as well as 3 other papers that have been published in 2018 to 2019. And these are some other papers that have been published so far from 2016. And I just hope that you enjoyed my presentation. Thank you very much. Thank you. It's very difficult to finish it so quick.